Chapter 2. Building Blocks of the Human Body. Life in multicellular organisms emerges from extraordinary structural and functional coordination among billions of cells. Although invisible to the naked eye, cells constitute the fundamental units of biological organization upon which all physiological processes depend. From the earliest stage of embryogenesis, a single fertilized ovum divides, differentiates, and orchestrates increasingly complex patterns of structure and specialization. The human body is therefore best conceptualized as a dynamic hierarchy. Cells form tissues, tissues assemble into organs, and organs integrate into systems that sustain homeostasis and enable survival. Understanding this hierarchy is essential for medical learners, because virtually every disease process can be traced to disturbances at the cellular and tissue level. Cellular architecture and biochemical governance. A typical human cell consists of cytoplasm enclosed by a selectively permeable plasma membrane and contains highly specialized organelles suspended within a cytosolic matrix. The plasma membrane, composed primarily of a phospholipid bilayer with interspersed proteins and cholesterol, functions as both a protective barrier and a regulatory interface between intracellular and extracellular environments. The polar heads of phospholipids face aqueous compartments while hydrophobic tails form a central lipid layer, creating selective permeability that permits only certain molecules to traverse passively or actively. Embedded proteins serve as receptors for hormones and signaling molecules, transport channels for ions and metabolites, mechanical anchors for the cytoskeleton, and immunological identifiers that distinguish self from non-self. Within the cytoplasm, organelles execute tasks fundamental to cellular vitality. The nucleus houses chromatin and directs gene transcription, protein synthesis, and cellular replication. Mitochondria generate adenosine triphosphate through oxidative phosphorylation, supplying metabolic energy to fuel muscular contraction, neuronal signaling, and biosynthetic reactions. Ribosomes translate mRNA into polypeptides, while the rough endoplasmic reticulum supports protein synthesis and the smooth endoplasmic reticulum facilitates lipid metabolism and detoxification. The Golgi apparatus packages proteins for secretion or membrane insertion, lysosomes digest intracellular debris, and the cytoskeleton maintains cell shape while enabling motility and intracellular transport. This complexity is not static. Cells cycle through periods of growth, DNA replication, and division. Mitosis yields genetically identical daughter cells for tissue maintenance and repair, while apoptosis eliminates damaged or senescent cells in a controlled manner without provoking inflammation. By contrast, necrosis results from injury, ischemia, or toxins and triggers inflammatory responses that may contribute to tissue dysfunction. Homeostatic transport and molecular exchange. Cell survival demands constant exchange of gases, nutrients, electrolytes, and metabolic waste products. Passive processes such as simple diffusion and facilitated diffusion permit movement down concentration gradients, whereas active transport and ion pumps expend ATP to maintain electrochemical gradients essential for nerve conduction, muscle contraction, and acid-base equilibrium. Osmosis enables water movement across semi-permeable membranes in response to osmotic gradients. Specialized transport mechanisms also support endocytosis, by which cells ingest extracellular substances through phagocytosis or pinocytosis, and exocytosis, which allows secretion of hormones, neurotransmitters, and enzymes. These mechanisms collectively preserve internal constancy and responsiveness in a changing external environment. Tissue organization and physiological differentiation. Cells with shared morphological and functional profiles organize into four principal tissue types: epithelial, connective, muscle, and nervous tissue. Their arrangement underlies the architecture of organs and dictates their functional properties. Epithelial tissue covers body surfaces, lines cavities, and forms glandular structures. Its tightly packed cells provide protection, regulate absorption and secretion, and participate in sensory reception. Simple epithelia facilitate diffusion and absorption, whereas stratified epithelia resist abrasion. Transitional epithelium accommodates expansion in organs such as the urinary bladder. Glands derived from epithelial tissue may secrete onto body surfaces via ducts or release hormones directly into the bloodstream. Connective tissue constitutes the most abundant and diverse tissue class. Its extracellular matrix may be fluid, gelatinous, fibrous, or mineralized. Loose connective tissue binds structures and provides elasticity, while dense connective tissue forms tendons and ligaments capable of withstanding tensile forces. Adipose tissue stores lipids and regulates metabolism. Cartilage and bone provide support, facilitate movement, and protect vital organs. Blood, a fluid connective tissue, transports respiratory gases, nutrients, hormones, and immune cells while participating in homeostasis and defense. Reticular tissue forms scaffolding for lymphoid organs, and elastic connective tissue maintains vessel compliance and airway recoil. Muscle tissue generates force and movement. Skeletal muscle produces voluntary movement and posture, smooth muscle regulates diameter of airways and blood vessels, propels contents along the gastrointestinal and genitourinary tracts, and contracts the uterus during labor. Cardiac muscle maintains rhythmic contraction and blood ejection through intrinsic pacemaker activity. Body systems, anatomical framework, and cavities. The human organism comprises 11 major organ systems that work cooperatively to maintain life: integumentary, skeletal, muscular, nervous, endocrine, cardiovascular, lymphatic, respiratory, digestive, urinary, and reproductive systems. The skeleton provides structural support, protects vital organs, and serves as a reservoir for minerals. Axial components include the skull, vertebral column, and thoracic cage, while the appendicular skeleton consists of the shoulder girdles, upper limbs, pelvic girdle, and lower limbs. Musculoskeletal integrity enables mobility, posture, and protection of neurovascular structures. The body is organized into major cavities housing and safeguarding organs. The cranial cavity encloses the brain, the thoracic cavity contains the lungs and mediastinal structures, the abdominal cavity houses digestive and accessory organs, and the pelvic cavity accommodates reproductive organs, bladder, and distal bowel. Serous membranes such as the pleura, pericardium, and peritoneum facilitate friction-free movement of organs, while synovial membranes lubricate joints. Mastery of anatomical terminology, directional descriptors, and planes of section enhances accuracy in physical examination, surgical planning, and diagnostic interpretation. Cellular adaptation and disease foundations. Health depends on continuous renewal, controlled proliferation, and regulated cellular differentiation. Physiological adaptations such as hypertrophy and hyperplasia support increased functional demands, whereas atrophy reflects decreased workload or impaired nutrition. When cell cycle checkpoints fail or genetic mutations accumulate, aberrant proliferation may occur. Benign neoplasms remain localized and preserve some differentiation, whereas malignant tumors invade and metastasize, representing a loss of growth regulation in cellular architecture. Cancer biology therefore embodies the consequences of dysregulated cellular mechanisms that normally preserve tissue integrity. Stem cells and regenerative potential. Beyond terminally differentiated cells, the human body harbors stem cells capable of self-renewal and lineage commitment. Embryonic stem cells possess pluripotency, able to generate all cell types, whereas adult stem cells, such as hematopoietic and mesenchymal stem cells, maintain tissue repair via more restricted differentiation pathways. Advances in induced pluripotent stem cell technology have transformed modern biomedical science by reprogramming somatic cells into pluripotent cells, enabling disease modeling, regenerative therapies, and precision medicine. Clinical translation continues to evolve, with ethical considerations and safety challenges including immune compatibility and oncogenic risk shaping future therapeutic applications.